measure synaptic currents very easily. Measuring synaptic currents in most parts of the brain, or in most nerve cells, involves sticking a tiny little microelectrode or micropipette inside the cell. The cell is tiny, and it's difficult to hold an electrode inside a cell for long periods of time. If what you're doing is looking for a neural model for memory, for looking for a Hebb synapse, you want a technique which will allow you to follow synaptic strength for long periods of time. And so this is exactly what one can do in a layered or laminated structure like the hippocampus, and you can't do it in other regions. So, for both reasons, because the hippocampus we know is involved in the formation of new memories, and because it's an easy structure to study, it seemed like a good place to look for Hebb synapses. I had just moved to Mill Hill at this point, and I was mindful of the description of science by Sir Peter Medawar, who's then the director. Science is the art of the possible, like politics, in fact. And here it's possible to do experiments and follow synaptic changes in synaptic strength for long periods of time. So, let's have a look at what I'm talking about here. So here are the granule cells, these cells down here. Here's the medial perforant path, that's the unstained region here. And those perforant path fibers just terminate on the middle part of the dendrites of these granule cells. And when you stimulate the perforant path and record with an extracellular electrode, that means to say you just have to put an electrode in this region, and you'll be recording from lots of cells, but they'll all be activated simultaneously. And because just this part of the membrane is activated, you'll see a current there. And actually the direction of the current depends, or of the voltage depends on where you are. But in the synaptic region, it's negative going. So that is a synaptically generated response. And actually so is this, but it's reflected in polarity. These are synaptic responses. No cells are firing. You're measuring synaptic current. You're measuring, in fact, the strength of that input. If you increase the intensity of stimulation and activate more and more of these fibers, eventually you'll excite the granular cells so much that they will fire. And that causes this downward going spike here, which is called a population spike, which is a reflection of the population of granule cells that's firing. And if more cells fire, that will get bigger. The actual nerve impulse itself doesn't vary. It's what's called all or none. But if you have more cells firing, then you'll generate a larger population spike. But from the point of view of the experiments I'm about to describe, this is actually the important thing. This is the synaptic current, because it's the synaptic current that we want to measure. Hebb is telling us that synapses change, so we want to measure synaptic current. So, at last, we were able to identify synapses which had this Hebbian property of changing their strength for long periods of time, following brief periods of activation. So we're not exactly modeling memory because what we're doing is stimulating axons intensely. But we're talking about a model of the learning process here, and we'll come back to its connection with learning later. So here is an experiment that we did in those days in Oslo. This is the strength or the amplitude of those synaptic responses. And the hippocampus is bilateral structure, and this was in experiments were done in anesthetized rabbits. And you can see that if all you did was just give test shocks of constant intensity throughout the period of this experiment, nothing changed very much. But if you gave high-frequency stimulation just for a few seconds and then went back to the low-frequency testing, there was an immediate and sustained increase in the amplitude of the response, in the amplitude of the synaptic response. And each time high-frequency training was given, it got a bit bigger. Eventually, it would saturate. So that's an example of a long-lasting change in synaptic efficacy brought about by a brief alteration in synaptic bombardment. And this is lasting a long time, it's several hours. And in those days, changes lasting for a minute or two were the most that people had routinely observed. You might be wondering about this gap here. I can tell you now that it's safe to do so, that at this point, both my colleague Terry and I fell asleep. And there were no computers in those days. So there's that gap. That's confidential information, by the way, because you never know with referees. They might demand that we rescind the paper. But they didn't actually notice it, and we weren't about to tell them. So here are some properties of LTP. This phenomenon, we called it long-lasting potentiation. And I don't know why we did, but I seem to remember Tarja Lermo, who was Norwegian and therefore spoke much better English than me, telling me that long-term potentiation was bad English and that long-lasting it should be. So we called it long-lasting, but then it became impossible to actually say or to produce an acronym of long-lasting potentiation. It became LLP, which is not something that trips off the tongue. So it eventually became long-term potentiation, and that's what we call it. And so LTP, or long-term potentiation, has these three very attractive properties as a memory model, as the basis for a neural model of memory. It lasts a very long time. I showed you lasting for several hours. In animals which have had electrodes implanted under anesthesia and then allowed to recover and then are just wandering around, freely moving animals, LTP has been followed for up to a year. So it's a very long-lasting phenomenon and may well last a lifetime. So it's long-lasting, it's input-specific. That means that if you've got two pathways into a cell and you give one of these high-frequency trains to one pathway, the other pathway will not be affected. So it's not some sort of change in the whole cell, in the whole target cell. It's a change which is specific to the pathways which have received this high-frequency stimulation. And that obviously hugely increases the potential mnemonic capacity of the neural machine. It's also associative, which is a slightly more complex thing, but if you have two inputs again, and one is too weak to induce LTP, and the other is a strong input which will induce LTP. If you just give the weak one alone, it won't potentiate, but if you give it together with the strong one, then both pathways are potentiated. And that provides an immediate model for things like classical conditioning. So it's got these neat and attractive properties, which make it very likely that it's involved in memory. I see that I'm running out of time. So I must now hurry along and say that one of the great triumphs of research in this area has been to explain the properties of associativity and input specificity on the basis of the actual protein, the actual receptor, which is required for LTP. And that turns out to be the NMDA receptor. That's the one which is normally blocked by magnesium. And in this famous experiment, it was shown that if you block the NMDA receptor, if you give a high-frequency train which would normally induce long-term potentiation, but block the NMDA receptor with this drug, this antagonist called AP5, then you don't get LTP. So you need the NMDA receptor to get LTP. If you wash AP5 away and give exactly the same train, now you get long-term potentiation. So we have a very good idea about how LTP is induced. Let me just skip that and simply say that there's an induction rule for LTP, which really says that whenever the conditions are right for opening up the channel of the NMDA receptor, and that channel is permeable to calcium, When calcium enters the cell, or enters the synapse, enters the spine through the NMDA receptor,